Hi everybody, welcome back. Coming to the end of chapter 1 here. And what we're going to look at here really is just to refresh your thinking about how we do science, and then take a look at some model organisms which have been used extensively to study cell biology. And the last thing we'll do is we'll finish up looking at some experimental design, and this will be useful for your data analysis problems that you have to complete every other week. So let's get going. One of the first things that scientists do when they start thinking about solving new problems or making new discoveries, answering new interesting questions, is they have to understand the current state of affairs. They've got to understand the background to the field. So let me give you my experience of this as a PhD student. As I was working on my PhD, I would accumulate papers, and I eventually had a stack of papers about yay big that I was trying to read through, just to catch up on the state of the field, so that I could understand what had been done, what we knew. There's no point in replicating other people's work. I mean, it's important to replicate that you can replicate other people's work, but that doesn't necessarily produce new knowledge. So that's really the first thing that scientists do, is they become expert in the current state of affairs or the current state of the field. And then they obviously make interesting observations about new phenomena, and that often leads to new questions. How is this happening? Why is this happening? What's the mechanism for this? And then they can either make careful observations about experimental systems or about the world around us, or maybe they do experiments, but they're gonna collect data, and they have to make very, very careful observations, either of the data they're collecting or of the natural world as they make observations of natural phenomena. So careful observation is critical in science, and so that's something to keep in mind. People have done experiments on all kinds of different types of organisms over the years, and have advanced the field of cell biology, using organisms all the way from simple viruses, even though they're acellular, all the way up to complex animals such as monkeys. So there's a whole range of model organisms that scientists can use to study cell biology. We'll take a look at 6 here, and it's just useful to have a general idea of what the important features of a model organism are. Generally, they're gonna reproduce pretty quickly. They're going to be easy to keep in the lab. They're going to be relatively cheap to keep. There's obviously a range, and mice are more expensive and more difficult to keep than bacteria are. But there are things you can do with mice that you can't do with bacteria, so there's a trade-off there. So we learn a lot about cell biology from studying bacterial processes. We've learned a lot about the mechanisms of DNA replication, DNA repair, all kinds of genetic mechanisms, and cell division processes. Probably the best thing about E. coli is that they're haploid. That means they've only got one copy of each gene. So if you delete a gene, you can pretty easily see whether it's essential or not. And you can put mutant versions of the gene back pretty easily. They're small, they're unicellular, and they grow incredibly rapidly. And they've also become obviously very widely used for genetic engineering, for cloning and that kind of stuff. So they're used as an organism to study cellular biology, but also as a molecular tool, really. A little bit more complicated then, we get into Schizosaccharomyces cerevisiae, and it has a relative, Saccharomyces pombe. So this is budding yeast and fission yeast, respectively. These have been used for many, many years. Obviously eukaryotic. Again, they grow very, very easily in the lab, very, very quickly. And there are lots of naturally occurring and man-made mutant lines which you can use to study gene function and therefore cellular processes. They were hugely, hugely important in the discovery of proteins involved in the cell division cycle. And a colleague of my PhD supervisor, a man called Paul Nurse, used yeast to make one of the fundamental discoveries that earned Paul and my PhD supervisor the Nobel Prize in 2001 for the discovery of a number of proteins that underlie the mechanisms of cell division. So that's E. coli and the yeasts there. And then we've got more complicated organisms, so getting into metazoans now, so that's more complex animals. Drosophila melanogaster, fantastic genetic model organism, pretty easy to keep in the lab, pretty cheap to keep in the lab. They eat yeast. So if you see these hovering around your fruit bowl or around your alcohol collection, they're after yeast that are growing around your fruit and around your booze. Lots and lots of genetic, a lot of mutant lines of the flies. They're pretty easy to mutagenize, so you can change their genetic background and you can do crosses. You can make transgenics, so you can knock genes in, you can knock genes out, that kind of thing. And they've been used in a lot of studies on embryogenesis and development and differentiation. You've probably seen the Antennapedia fly that's got these two legs coming out of its head. It's kind of funky. And on the right there is a roundworm, C. elegans, Cinerhabditis elegans. Probably the coolest thing about C. elegans is that when it starts out as a fertilized egg and then develops into the full-grown worm, we know the fate of every single cell in the lineages that end up making up the adult worm. So you can look at a cell, and before it divides, you can say, okay, the offspring of these cells are going to end up being in the gonad or in the olfactory receptors, those kinds of things. So it's been used to do a lot of cell fate mapping and lineage mapping, that kind of stuff. More complex still is the mouse, Mus musculus, the house mouse, and also used widely as Rattus norvegicus, the Norwegian rat. And so, great thing about this is that it's very, very similar in terms of cellular processes to humans. There obviously are differences, and there's a lot of controversy about how reliable mouse studies are in terms of application to human biology. But there are lots and lots of similarities. We're obviously not too far distantly related from them, phylogenetically speaking. Again, it's become pretty easy to knock genes out. To knock genes in, so you can knock a gene out and then you can add in knock in a mutant form of the gene and look to see what effect that has on cellular behavior or phenotype, that kind of thing. You can even make conditional knockouts, which is where you've got a gene which is going to be lethal during embryogenesis if you take it away. So if you knock the gene out in the mouse, and it's important during embryogenesis, you obviously don't get a viable mouse. And so what you can do is you can make a transgenic mouse where the gene gets inactivated only when you treat the mouse with a certain drug, that kind of thing. So you can look at certain genetic defects only in the adult mouse rather than killing the embryo. You can turn on fluorescent proteins in the mouse, in the ears, and all kinds of kind of trickery like that you can use. So that's the mouse, and then finishing up there with Arabidopsis thaliana, that's a mustard, also known as stale cress. If you did General Biology 1 at UC Denver, then you used a relative of this in the flask when you did the flask plants experiments. Very, very easy to grow, rapidly cycles, lots of mutant cell lines. And obviously used to study plant development and that kind of thing. So those are some model organisms. Think about the fact that these are all relatively easy to keep in the lab. Some more so than others, obviously. If you're working on yeast and they don't get up and run away or fly off, so that's a good thing. They're cheap, and they're relatively easy to work with at the genetic and the cellular levels. So think about what makes a good model organism. Monkeys, for example, great because they're closer to humans phylogenetically, but on the other hand, lots and lots and lots of problems with keeping monkeys in terms of the husbandry, keeping them alive in captivity, and then also with the regulations around using animals which are more closely related to us than the little mouse there. I'm going to put cell culture in here as a model organism. Obviously, it's not an organism per se, but there are hundreds of cell lines which have been developed for the use in labs all around the world. Probably the most famous is a cell line called HeLa, derived from a cervical carcinoma of a lady called Henrietta Lacks. And if you're interested in that story, it's described on page 16 of your textbook. There's also a whole text. There's also a whole book about Henrietta Lacks and her story and the story of her family. So these cells can be grown in dishes, and so you put the cells in these specially treated dishes, and you give them a nutrient media, a nutrient broth like you give bacteria, so it's a nutrient media, and then they either grow in suspension, so they float around, or they stick to the bottom of the plate, and that's what you see on the right there. Those cells are all stuck to the bottom of the plate, and then you can study the effects of adding genes. You can look to see where genes localize, that kind of thing. The problem with these cell lines, though, is that they've changed so much. They're genetically so different from their original tissue. They obviously don't behave like a tissue. They don't form tissue-like structures, not always anyway. They can be induced to form certain structures under certain conditions, but they may be very, very kind of different in terms of the genetic background if you compare them to where they originally came from. So there are some caveats and problems with using cell culture, but they're used widely to study cell division, cell differentiation, cancer biology, the mechanism of actions of certain viruses and how viruses replicate, the behaviors of certain proteins under certain conditions and that kind of stuff. So they're an invaluable research tool in cell biology. Now, you won't find this in your textbook. There's a little URL. I've put a shortened URL on this slide. If you want to see a little bit more about 3D printing, you can type that into your browser. You can't click it on anything, but it's pretty easy to type into your browser. So I've thrown this in here under model organisms as well. There have been huge advances in 3D printing. You've already heard about 3D printing in the popular press. This is where you have a printer which basically prints three-dimensional structures. And it's evolved to the point where you can print a matrix onto which you can then overlay cells, and those cells under the right growth conditions will actually form the tissue. So on the left, there's a jawbone. That's a piece of jawbone that can be used in transplant. On the right there, you can tell that's an ear. So the connective tissue has been laid down using the 3D printer, and then that's been overlaid by layers of cells, which have then differentiated into the structure of that ear there. So that link down there is an ABC story about 3D printing of tissues at Wake Forest University. So if you're interested in that, you can take a look at that. Okay, so let's finish up this with a quick look at experimental design and how we think about doing experiments. Some of this is going to be probably pretty obvious and familiar to you guys. When you do an experiment, you want to change one variable at a time. That means that you can see the effect of the changed variable on the outcome of the experiment, if there is any outcome. So if you change variable A and you see outcome B, then you're a bit closer to having a cause-effect relationship. Obviously, you can't say there is a cause-effect relationship, but you're closer to getting that. If you change A and C, and then you see that B doesn't change, you don't know whether A caused B to go up and C then caused it to go down. It becomes much more confounded and complicated. Obviously, when you're doing an experiment, you want replicates. So if you are treating cells with a drug which you think is going to cause the cells to die, you want to do that multiple times so that you don't just do it once and you happen to have an outlier and then you make a conclusion based on an outlier. And then the experiment has to be repeatable. So repetition is different from replication. Replication is when you have 6 duplicates of a sample, and then you look at your samples and you take an average of the samples and you calculate a standard deviation of the mean and that kind of thing. Repetition of the experiment is that you do the experiment once and you collect your data together and you reach a conclusion, and then repetition is, can I do the same thing again and get the same results? Can someone independently do my experiment if I give them the protocol? So this is why when you look at research papers, they have these materials and methods, and the goal is to have people be able to repeat the experiment and get the same finding. If the findings are truthful, then you should be able to repeat the experiment ad nauseum and get the same conclusions. So let's take a look at a couple of pieces of data. This is data from a paper that I gave my 4,000 level cancer biology students as part of their finals. So they were given the paper. They weren't given any of the text in the paper, or the introduction or the abstract, all that kind of stuff. And they had to just look at the figures from the data and reach the main conclusions of the paper. So let's just take a couple of looks, a look at a couple of pieces of data from that paper and think about some of the things that you'll have to think about when you do your data analysis problems in Canvas. So as you look at this, the first thing is that it's overwhelming. There's so much stuff going on here. So on the left is the actual data, and that's data from a number of experiments. It's been plotted on a graph so we can quickly interpret the data. On the right, I've just put down some information about the experiment so that you have it if you come back and you want to look at this again. You've got some text for this. So what's being done here is a cell line called U2OS has been grown in vitro. So that just means in glass, it means in dishes. So the cells are growing in dishes. Just for interest, if you're interested, U2OS cells were derived from an osteosarcoma. So that's a connective tissue cancer. So these grow very well in dishes. They don't have any problems. You give them the right nutrients and they grow very nicely. And the investigators here are interested in this protein's effects on the cells in the presence of this drug called MMC. So MMC is mitomycin C, and that's a drug which causes DNA damage. And so when you damage DNA in cells, if the cells can't fix it, then they cause themselves, they trigger a process called apoptosis, so they cause themselves to die. And so what we're looking at here really is the effect of this drug mitomycin C, and the concentration and the dosage here is in nanograms per mil. So we've got these different dosages, so there's different types, different treatment levels. So there's the treatment level down here on the x-axis. The drug, remember, is mitomycin C. That causes DNA damage and it's going to cause the cells to die at certain concentrations. And then we're measuring how many of the cells die. So here we've got 100% cell survival, so obviously no cells die. And then down here, only 0.1% of the cells died. 1% of the cells, sorry, only 0.1% of the cells survived. So that's 99.9% dead cells. 1% cell survival, that means 99% of the cells die. So that means that most of these cells are dying. So then we've got a number of lines on the graph. So let's take a look at these lines. The gene that the authors are looking at is a gene called FAN1, and it's involved in DNA metabolism. It's involved in protecting cells against DNA damage. That's why they're interested in it, and that's why they're using it. They're looking at the effects of this drug on the cells. So when it says FAN1++, that means that the FAN1 gene, and therefore the FAN1 protein, is present in the cells. So they have a neat genetic trick they can do to remove both copies of the FAN1 gene. So ++ here in these brackets and over here on the left means that both copies, that both alleles are wild type, so they're working perfectly fine. -- means that both alleles in the diploid, obviously these cells are diploid, they're from humans, both alleles have been deleted. So that's a null. So there, the gene has been deleted completely and there's no protein produced. So let's just take a look at that data to start with. So that's the circle on the graph and the square on the graph. So let's look at the circle. So what you see is that as the concentration of drug goes up here from 0 to 20 nanograms per mil, when FAN1 is present, that's the circles there, even with 20 nanograms per mil of mitomycin C, you're down here at about, I don't know, 25 to 30% cell survival. Now, if we come down and look at the square, which is FAN -, so that's those cells don't have the protein, the FAN1 protein. We're now down, if I go over to this axis, that's about 2%. Note this is a log scale, so we're going from 100 to 10 to 1 to 0.1. So this is a log scale. So the effect of deleting the FAN1 gene is you get much greater sensitivity of the cells to this drug mitomycin C. So that means if the gene is gone, you give the cells 20 nanograms per mil of mitomycin C, they're 10 times more sensitive to it. And so the conclusion would be there that the cells probably can't repair the damage caused by the mitomycin C when they don't have FAN1. So FAN1, this tells us FAN1's got something to do with repairing the DNA damage caused by this drug. So then there's another couple of experiments down here. So those are 2 experiments to start with. There's another experiment down here as well, another 2 experiments down here. The triangle is the FAN1 null mutant. That means both alleles have been mutated. And then they've used a genetic trick to add FAN1 back in again, so they can put the gene back. So they delete both copies and then they add it back. And so they're just trying to check that their process of deleting the gene isn't responsible for this sensitivity that they see over here. So they're just putting the FAN1 back, and that's the triangle. So when we put the FAN1 back, we go back to the kind of wild type levels of sensitivity. Now, if we put in this FAN1 UBZ star instead of just normal FAN1, what happens then? So that's the kind of black filled in hexagons. Now you can't see it, but they're buried under here. So they're buried behind here. They run very close to this one here. So when you put that mutant in, you get the same kind of sensitivity to the drug as you do if you just don't have the gene at all. So this version of the gene here doesn't help you rescue the phenotype of the sensitivity to mitomycin C, because we still see the sensitivity here. So what is this mutant here? That mutant there is a mutant in a domain of the protein which helps that FAN1 protein bind to other proteins involved in the repair process. So even though FAN1 is there, it's got a mutant in this domain which it's using to bind to other proteins to help fix the DNA. So so what this tells us is this UBZ domain is really important in the function of FAN1. So there are a lot of experiments going on here, but they're all being presented on one piece, on one data, on one plot, or one graph, I should say. So the important thing here is there's only one variable being changed. Okay, the variable being changed is whether FAN1 is present, FAN1 is absent, or FAN1 is absent and being put back in, FAN1 is absent and the FAN1 UBZ mutant's being put in. So what we have is we have that variable that's being changed. We are then measuring cell survival as a function of mitomycin C concentration. So what is the dependent variable here? The dependent variable is what we're going to measure. So that's cell survival. Cell survival is dependent on the genotype of the cells and the presence or absence of mitomycin C. So this treatment level down here of 0 nanograms per mil, all of the cells survive. But as we change the treatment level and we increase the concentration, we start to see these differences between the FAN1 wild type and the FAN1 null, and the FAN1 wild type and the FAN1 null that's had the FAN1 UBZ mutant put back in. Okay, so you got to think about what's the dependent variable, what's being changed, what's being measured. Am I looking at multiple experiments? Okay, there are multiple experiments being shown here. Okay, so that's one piece of data. Let's take a look at another one. Same kind of thing is going on here. We're still dealing with this FAN1 protein, but now we're looking at the effect of this FAN1 protein in a cell line called MEFs. MEFs are mouse embryonic fibroblasts. So these are fibroblasts. They're a type of cell that they're isolated from mouse embryos, so they're called mouse embryonic fibroblasts. And what we're looking at are MEFs either with FAN1 present. So FAN1 present would be the FAN1 genotype down here plus plus. So that's both alleles are present, FAN1 plus plus. Not present, that would be the minus. If you see minus minus, then that would be FAN1 not present. Now we're not actually seeing any of that down here. Is what we're seeing is this ND. FAN1, part of its job is as a nuclease, and so a nuclease is something which can act on, can break nucleotides off DNA or cut DNA. And so ND means nuclease deficient. So that means you've deleted the nuclease ability of the FAN1 protein. So what we've got on here, the FAN1 genotypes we're looking at are plus plus, so that's wild type, and then nuclease deficient. Basically, you can think about that the same as being minus minus. Functionally, it's like minus minus. It's like the FAN1 mutant where there's no FAN1 produced. FAN1's being produced, but it can't do its job because its nuclease domain is nuclease deficient. So that's the genotype down here. And then there are two levels of treatment here. Mock treatment or treatment with that drug again. So rather than doing a range of drug treatments, they've picked a drug concentration. It's probably about 15 or 20 nanograms per milliliter. And then they're either just adding whatever the drug's dissolved in, that's the mock treatment, or they're adding the drug. And they've been kind enough to split this into like a white and a gray area. And so one of the comparisons we want to do is between this side of the graph and this side of the graph. So we've got two treatments, no treatment at all, or no drug, 0 drug concentration, that's one level of treatment, and then another level of treatment over here. Now remember, this drug causes DNA damage, and if FAN1's not working, then we are going to end up with a lot of cells dying. Now, is what we're measuring here the dependent variable here, what we're going to measure, is not cell survival this time, but it's chromosomal abnormalities. So they're looking at these cells microscopically, and they're able to see the chromosomes, and they're looking for certain types of chromosomal abnormality. And you can see these down the light microscope with certain preparative techniques on the cells. So they're plotting the whole range of data for us, and then they're putting in the mean with the standard deviation shown by the bars going up and down. So that's the kind of graph there. What is this FAN1 virus business here? So in the last graph, I'd said they've got this genetic trick for adding the genes back in again. So they can add in the wild type gene, or they can add in this gene that's got the mutation in the UBZ domain. Remember, that's the domain which causes other proteins to associate with FAN1. So the way they're adding it back in is they make a virus which contains the FAN1 gene or the FAN1 mutant gene with the UBZ domain mutated. So that's how they're getting the FAN1 back in again. So let's just take a look at this one piece of data over here on the left. So the cells, the MEFs, are wild type for FAN1. They haven't put any FAN1 virus in. They don't need to because the cells are wild type. So these are probably just perfectly normal mouse embryonic fibroblasts. And they haven't treated them with mitomycin C, and so there's a very, very low basal level. So at this level of treatment, no treatment at all, there's a very low level of chromosomal abnormalities. Now let's go over here and look at this one. So I can compare this one and this one. FAN1 plus plus, no FAN1 has been added back in. And when we treat with mitomycin C, we see that there's an increase here relative to here. We see there's a slight increase. We don't know if that's significant or not, but there's a slight increase. So, is what we're really going to look at then is are these three lanes here and comparing them with these three over here. So the FAN1 genotype, nuclease deficient. So this is effectively FAN1 that doesn't work at all. And it's the same thing over here. So if you recognize this whole thing on the left is duplicated on the right. So this is effectively no functional FAN1, and then added back wild type, or the wild type but with the mutant UBZ domain. So now, what happens if we do the same thing that we add mitomycin C? So there's a treatment. We've got a treatment level of mitomycin C, let's say 15 nanograms per mil of the drug. Okay, so if we don't have FAN1, if it's nuclease deficient in the genotype, and we don't add FAN1 back in, we see a pretty big increase. So we can compare this row, this column on the graph here, to this one over here. And the authors are saying that's really significant, okay, because we've got a big difference between the mean here and the mean over here, and the standard deviations are sufficiently small. If we have nuclease deficient FAN1, and then we add back in the wild type FAN1, we can drop this level of chromosomal abnormality back down to something that was comparable to what we have when we have wild type FAN1 in the cells. If we mutate the UBZ domain and we add that back into cells which have got the nuclease deficient FAN1, then we see a similar increase in the chromosomal abnormality number than if we don't add FAN1 back at all. So, dependent variable. Chromosomal abnormality is dependent on two things here. Really, it's dependent on the genotype and on the level of treatment in terms of whether mitomycin C is present or not. Okay, so these data are going to be complicated, but you're going to have to break them down and look at them carefully. And I would really encourage you to work on this with a classmate. 2 brains, 3 brains are better than 1 on this. Now you've got to turn this in on your own. It said I was a quiz in Canvas, but if you want to, you're more than welcome to work with 2 or 3 other people on this and put your heads together on it. So we'll wrap up there, and next time we'll start in on chapter 2.